Today's speaker is Dr. Anna Lindholm from the Department of Evolutionary Biology and Environmental Studies at the University of Zurich, where she's an independent group leader. Anna's interests are behavioral ecology, ecology, evolutionary genetics, cooperation, selfish genes, social behavior, and social selection. She studies house mice in the wild and is integrating behavioral, ecological genetic, and genomic studies to understand the evolution of social behavior. The mice Anna studies harbor a male meiotic drive system, and we're going to hear about some of the interesting results she's obtained regarding the impact of the system on behavior. Anna, thanks for participating in our webinar series. We're really looking forward to your presentation. Let me turn the microphone over to you.
Thank you so much for the invitation. So I'll be speaking today about work that I've done in collaboration with quite a large group of people on a natural gene drive system in house mice. And what interests me are the frequency dynamics of gene drive systems in nature and what factors contribute to them. When I mean gene drive, I'm talking about inheritance patterns that are greater than Mendelian expectations. So from this figure, if we think about the red allele on the left, under Mendelian expectations, it's inherited to 50% of offspring. But under drive, it's inherited to a greater proportion of offspring. In this figure, 100% of offspring inherit the allele from the parent. And so I'll be discussing a system which shows drive. And we found one by accident in an intensively studied house mouse population in Switzerland. So this is a study that was started in 2002 by Barbara Koenig and Andrea Veit, her PhD student, and they were interested in studying cooperation in house mice. And so they set up a study site where they caught mice locally, brought them into this old building, and then tried to make it comparable in some ways to how commensal house mice live. So in Central Europe, commensal house mice live with people and animals, so they're feeding on spilled feed and they get water from sources usually provided for animals or so, and they have hiding places to nest. So we've provided them with nest boxes where they can nest securely. We provide them with food and water and hiding places, and they live here voluntarily. And this study has now been going on for almost 20 years. And interestingly, this building is quite permeable to dispersal and immigration. So they can come and go as they please. And if they don't want to live here, then they can leave. And what we found out subsequently is that some of these founders that were brought into this population carried the t-haplotype. That was not known at the time. It was only because DNA samples from these mice had been archived that we were able to find that out. And that led me down the path of investigating the t-haplotype. So what is it? It's a set of genes linked together on chromosome 17, an autosome in house mice. It takes up about half of the chromosome, and it has hundreds of genes on it. What does it do? Well, it transmits itself from male heterozygotes to 90% of offspring. So male heterozygote mated with a female wild type, 90% of those offspring will inherit the t-haplotype. And we know quite a bit about how this works. So here's a cartoon of sperm development in the testis. So we have diploid primary spermatocytes that divide into diploid secondary spermatocytes, and they then become the haploid spermatids. And evidence suggests that meiosis occurs normally, and we have 50% T allele carrying spermatids and 50% wild type carrying spermatids. And these are connected by cytoplasmic bridges these spermatids that descend from the spermatocytes. And that allows proteins and other materials to diffuse throughout these developing sperm cells. And the T haplotype has apparently taken advantage of this because it produces gene products that are spread throughout these developing spermatids, and they harm the wild type developing sperm. And they themselves carry a local antidote which protects them from this damage. We know quite a lot about the molecular biology of this system, thanks to work by Hermann and Bauer. And we know that there are at least 4 driver loci in different inversions, in different parts of the inversions. And they act upon a target locus called the sperm motility kinase, and that affects downstream motility, so that sperm that carry the T haplotype in the end swim more rapidly than wild type sperm. And because of this motility advantage, they are better able to fertilize eggs. And so they have effectively a sperm competition advantage within the male. And that's how they achieve this high degree of transmission. I think I mentioned that there are hundreds of genes within the T haplotype. And what do we know about what other genes do there? Well, in general, the T is enriched for MHC protein complex genes because most of these are on chromosome 17 within the region of the T haplotype. We carried out an RNA-seq study to compare gene expression between brothers that carried the T haplotype or just wild type alleles. And we found that 434 genes were differentially expressed, and less than half of these were actually on the T haplotype. So the T haplotype appears to be affecting gene expression elsewhere in the genome. And what is it that was being differentially expressed? Well, primarily genes that influence sperm development. So this fits our general model of how the T haplotype works. And this was the case for genes if we analyzed separately for genes on the T and genes off the T. So there's seems to be more to be learned about how the T affects sperm development. Okay. In females, the T is transmitted to 50% of offspring. So the T is affecting the testis and sperm development. In females, it is not biasing inheritance. So 50% of the offspring from a T heterozygous female mated to a wild type male inherit the T. But the T carries a recessive lethal. So when heterozygotes mate, then about half of the offspring inherit a T from each parent, and they die as embryos. And so the litter size is reduced by approximately half. So this is one of the fitness costs associated with the T haplotype. And why do these die? Well, offspring that inherit 2 copies of the T haplotype get 2 copies of a damaged gene that interferes with development. And that means that the T haplotype can never replace the wild type. It'll never fix in populations. So it can only exist as a polymorphism. There is little recombination in the T, and that means that deleterious mutations can accumulate within the T haplotype, and it's very difficult to get rid of them. And this probably explains the appearance of these recessive lethal alleles. Recombination has been studied to some respect by Rebecca Kellerman. She has been analyzing genomic data from a collection of wild type and T heterozygous wild-caught males. And she found that across the T haplotype, there is variation in how heterozygous T carriers are compared to wild types. So in some regions, there are strong differences between the T haplotype SNPs and the wild type ones. And in other regions, it's much reduced. suggesting that there probably has been some recombination in the evolutionary history. What Kellerman also found was that there is a high number of T-specific transcripts. So in the gene expression data, she found that there are differences in the sequences of the genes that were expressed in T heterozygous males compared to wild types. And these are not evenly distributed, but occur particularly in the proximal part of the T haplotype. And some of these T-specific transcripts were investigated a bit more carefully, and Kellerman found that some of these genes that have these T-specific transcripts appear to be duplicates of genes elsewhere in the genome. So these two here, PPP1CB and RNPEPL1, have copies on other chromosomes that are expressed in both T haplotype carriers and wild types. But there's a T-specific variant with a sequence difference that is expressed only in the T heterozygotes. And we tested these on mice from the barn study. And we don't know what function these might have on the T haplotype. So that will be something that we'll have to try to figure out in future. A little bit more about the origin of the T. There are these series of inversions. The second one is thought to have evolved first, about 2 to 4 million years ago. Mus spretus also has this inversion. And T variants between Mus musculus subspecies, because it's found in all of the subspecies, have relatively low variation, suggesting that one T variant swept through all the subspecies. But the recent work by Kellerman suggests that there's a bit more of a complex history. So that's something that we hope to find out more in the future. So back to the barn. So in our study, we can intensively monitor house mouse reproduction. So we can access the nest boxes that we provide. We can lift off the lids and look for the offspring and examine the offspring and take tissue samples from the offspring. And we get a lot of them. We get a lot of pups. So we have up to 2,500 pups a year. We get a tissue sample from all of them. We do a PCR test to diagnose the T haplotype. We can identify individuals by genotypes at 35 microsatellite loci, and we can do parentage assignment. And so with this data, we can look at changes in T haplotype frequency over time. And at the beginning of the study, 4 out of the 12 founders carried the T. And this plot shows the T frequency in pups born in 6-month intervals from the beginning. So at first, the T frequency remained fairly high, but then it dropped. And in 2011, we had the last pups born that carried the T haplotype. So it went extinct in this population and has not returned. And so we've been putting a lot of effort into understanding why these dynamics have occurred. So first of all, we asked, what would we have expected to have happened given 90% drive and TT lethality? Well, modeling suggests that the T frequency should remain high. So there's certainly more here that in this story than just drive and lethality. Something else is affecting the frequency. We know that increasing homozygosity disadvantages the T because of the recessive lethal. So anything that increases homozygosity could act to decrease frequencies. So inbreeding, for example. And we tested inbreeding using the microsatellite markers and found no decrease in FST values. And we're now looking at more subtle genomic measures, and it'll be interesting to see what we find there. But we have no indications that inbreeding has caused the decline. We've looked at natural selection, so is differences in viability between heterozygotes and wild types. And we found that females have higher viability. So this is a survival plot, and with t-haplotype here in gray, so that the t-haplotype heterozygotes in females had higher viability. So more of them were alive at a given age compared to wild types. In males, we didn't see a difference. So they seem to have an advantage. And that then is unlikely to contribute to T frequencies going down. And we tried to figure out a little bit more about why it is that females might be living longer. And we did a study of basal metabolic rates. And here's a plot of what we found. So here's mass on the bottom and resting metabolic rate at thermoneutrality. And in males, genotype made no difference. So the increase, the mass specific resting metabolic rate was the same for T carriers and wild types. But in females, we have quite different slopes. So it could be that there is something metabolically different between T heterozygous females and wild types, but we don't know what that could be. So having looked at natural selection, we then turned our gaze to sexual selection and competition for mates. So if you remember the results I showed you for inheritance of the T, when T heterozygous females were mated with wild types, they had a larger litter size than when they were mated with T heterozygotes. I've measured this in the lab, and a T heterozygous female on average gets 3 more living pups out of her litter if she mates with a wild type male compared to another T carrier, because of the mortality caused by this recessive lethal allele. So that could mean that there could be an advantage to females in general for mating with wild type males. And so we looked at this. So we did a series of preference tests. This is one in which we had a door and cage system. So these boxes here represent normal mouse cages, not closed boxes. So we had a female in the middle of either genotype and males confined to cages at the end. And so one male was a T heterozygote, the other male was a wild type, randomly assigned to these end cages. And they were all implanted with transponders. And we had a door system set up so that when a female approached the door, her transponder would be red and the door would open, so she could pass through. But for males, their transponder was red and that closed the doors, so they were confined to their end cages. But that meant the females could come and go as they wanted, and they could choose where they spent their time and who they mated with. What we found there is that there were no significant differences in relative time spent with males of each genotype. And when we looked at who fathered her offspring, we found no evidence there that this was due to differences in preference for males. What we found was that females often mated with both males. So we did other tests as well. We looked at whether females in estrus, if they were given a male to mate with of either genotype, how would they respond when they were given a second male to mate with afterwards? So if females have preferences for wild type males, they should mate more quickly with them, or mate longer, or refuse to mate with T heterozygous males. But we found no differences at all. And we also did a study using a preference box apparatus where females could choose to spend time with males of either genotype. And there we also found no evidence of a preference. And this is quite interesting because one of the few pieces of evidence of behavioral responses to the presence of selfish genetic element in the population comes from house mice. So Carol Lennington in the 1980s and 90s found evidence of preference for wild-type males. And we can't replicate that. So in our system, we see no evidence of preference. And we looked at sperm competition, because sperm competition, given that the t-haplotype affects sperm, is a promising avenue. And Hagen Bergstrom proposed that females could protect themselves from the effects of driver males, of reducing the risk of passing on driving elements to their offspring simply by mating with multiple males. Because if there is a sperm quality difference, so that sperm from driver males have reduced quality compared to wild-type males, then simply by mating with both, females increase the probability that wild-type males will fertilize her eggs, because they should have higher quality sperm. And in terms of the t-haplotype, we expect that half of the sperm from a t-haplotype male are going to be weak swimming wild-type sperm, and half of them are going to be comparatively normal swimming T-sperm. So if a female mates with both genotypes, she will get twice as much normal swimming sperm from a wild-type male compared to the normal swimming sperm she gets from a T heterozygous male. And that will increase the chances of fertilization by a wild-type male. So we did experiments in the lab to test this. So we took a female in estrus and mated her with either a wild-type male or a T-carrying male, and then introduced a second male and allowed her to mate with the second male. And then we assessed the paternity of the offspring before the offspring, before any of the offspring died from the lethal effects of, if they had inherited a copy of the T from the father and mother, because we assessed both genotypes of female. And what we found was that regardless of female genotype, regardless of mating order, T heterozygous male gained paternity of only 13% of embryos. So they're very poor sperm competitors. And in a completely different system, in experiments by Renee Furman and Lee Simmons, they also had a study system where they didn't know that they had the T haplotype present, and they found it out later. And when we reanalyzed their results in terms of whether the animals had carried the T haplotype, we found in their experiments that 19% of embryos were sired by T heterozygous males when they were in competition with wild-type males. So they're very poor sperm competitors. And so when we think about how the T haplotype works, I think instead that the T sperm are partly damaged. They're not immune, they're not fully rescued, but partly damaged. So we did a study recently looking at sperm motility. And here we compared sperm motility parameters from males in red carrying the T haplotype or in blue just wild type. And so we looked at standard measures of sperm motility and found in all cases reduced motility of T haplotype sperm for path velocity, straight line or progressive velocity, so how quickly they move forwards, for straightness, and for linearity. The other parameters of track speed and lateral amplitude and beat frequency did not differ. But what's interesting is that we don't see here evidence of bimodal distributions. So we don't see one bump for for fast sperm and 1 bump for slow sperm within t-haplotype carrying males. So it looks like that all the sperm from the t-haplotype carriers might be somewhat worse in their motility than sperm from wild-type males. And we redid this analysis looking only at the fastest sperm from these genotypes, and we found the same pattern. So if we compare fast sperm or sperm, there's a reduction in motility. And it's not quite fitting our expectations of T sperm being fully motile. We also looked at morphology. And on the left here is a picture of a house mouse sperm. So we measured midpiece length and tail length, and we looked at head length and head width. And we found that in red, again, the t-haplotype carriers the sperm have shorter tails on average and narrower heads. Whereas head length and midpiece length didn't differ. So there are morphological differences suggesting that there really are some morphological features associated with effects of the T on sperm. And when we compared these traits that differed against progressive velocity, we found that those with shorter tails had reduced velocity compared to longer-tailed sperm. So in blue, again, we have the wild-type sperm, and red, the T-carrier sperm. And you see there's not really very much overlap in terms of tail length. For head width, it wasn't so clear, and there we did not find a significant effect of head width. So there seems to be some morphological differences, and they might be explaining some of the effects on motility. And does it matter? It probably does matter. We find in the barn in our study population that T heterozygous males have reduced lifetime fitness. So in this graph, we have lifetime reproductive success on the y-axis and the average number of sperm competitors on the x-axis. In red, we have the T haplotype carriers, and in blue, wild types. So when there's no sperm competition, so females are mating monogamously, or at least they appear to, as in the litter, there's only one father, then genotype doesn't matter. They do equally well. But once there is sperm competition, then T carriers do worse than wild type. And on average, they have lower fitness in their lifetime than wild types. If we think about females, the story is a little bit different. So if females mate once, and if their genotype is T heterozygous and they mate with a T heterozygote, they get a small litter size. But once they mate with 2 males of each genotype, they get almost their full litter size back. And that is because the T heterozygous male sires so few of the offspring that almost none are lost to the recessive, get 2 copies of the T haplotype. And we find that in the barn, females mated with 2 males had the same litter size as wild-type females. So as soon as they mated with or had offspring fertilized by 2 males, they had equal reproductive success to wild-type females. And if they had only one father, they did a little bit worse. We observed multiple paternity in 47% of litters, and we've estimated that multiple mating occurs in about 61% of litters because males can mate with females but not produce offspring. So we've tried to estimate the missing proportion. So 61% is the best estimate. And there's no realized fitness costs on average to T heterozygous females. They have the same litter size at weaning as wild-type females and the same number of litters in their lifetime. When we add sperm competition into the model for T frequency changes, that then leads to a decline in T frequency. And of course, it depends how sperm competition is modeled. The higher intensity of sperm competition leads to a quicker decline in frequencies. But it does seem to play a large role in T frequencies. We also found that multiple paternity rates are related to population size. So these are adult population sizes at different times in the population. And the frequency of multiple paternity significantly increases with the adult population size. And modeling has suggested that T should be eliminated from the population when multiple mating rates are between 50 to 70% of litters. We also have evidence from a different t-haplotype system that enforcing polyandry reduces T frequencies. I mentioned earlier that Renee Furman and Lee Simmons had a study on house mice where they also didn't know they had the t-haplotype in the study at the time. So what they did is they did an experimental evolution study where they had lines where females were mated with a single male in each generation, and they had lines in which females were mated with 3 males in each generation. So the 3 male line is the polyandrous line, and the single male is here in green, the monandrous line. And over generations, the T frequency declined in those lines where female multiple mating was enforced. So we find in this different system also that sperm competition reduces T frequencies. So female multiple mating, we have evidence that it reduces T frequencies. It also reduces female reproductive costs associated with the T. And this, of course, depends on the T frequency, because the reduction in female costs comes when she mates with at least one wild-type male. And if T frequency is high enough, then she would be unlikely by random mating to mate with a wild-type and a T male. So we looked at whether the female multiple mating rate might differ between the genotypes because the benefits seemed to be higher for T carriers, but there was no difference. Moreover, the heritability of multiple mating was estimated to be about 0. So there isn't much potential for evolutionary change in female multiple mating rate, it seems. And this is quite different from studies in Drosophila, which have shown that female multiple mating can increase in the presence of a driving element. So multiple mating in this system is probably not an evolved response to the T. We also found that population density increased over time. So this is not quite up to date here, but from the beginning of the study, population sizes have increased quite a lot with males in black and females in red. And as the house mouse population grew, we also had a reduction in T frequencies. And this somewhat fits earlier results as well by Ardley and Silver when they did surveys of field populations of house mice to estimate frequencies of the t-haplotype in relation to population size. So these are population size estimates on the x-axis and frequencies of T heterozygous mice on the y-axis. And they found that large populations in general had either 0 frequency of the t-haplotype or rather low frequencies, whereas small populations were more likely to have high frequencies. So this fits very well with other studies. interestingly, we found another effect at high density. So studying in the barn, we had estimates of which pups were born in the population and which ones were still in the population as adults. And so the ones that were no longer present as adults had either died or emigrated. And whenever we find dead pups or dead individuals, we take a genetic sample to try to identify which animal that was. And so from this, we could see that there was no difference in mortality rate for heterozygous mice compared to wild-type mice, but there was a bias in disappearance. So T heterozygous house mice were more likely to leave the population before adulthood than wild-type mice, and this increased with population density. So this work was done by Jan Runge, and he wanted to know if it made sense that T heterozygotes should have higher dispersal. Would we expect this to evolve or not? So he carried out some simulation modeling, incorporating the known effects of the T haplotype of the sperm competition disadvantage and TT lethality. And he found that in his simulation models, dispersal evolved to be higher in T heterozygous individuals. So it does, so the increased dispersal is supported by this theoretical study, and also we tested it experimentally. So we created replicated populations at high and low density in small enclosures, and these were meant to represent kind of mini barns, about 7 square meters each. And we provided mice with a way out, a water barrier. So it was, and mice did not readily swim across this barrier. And we had across the other side of the water barrier a cage where mice could obtain food and water. And if they crossed the barrier and remained in this cage at the end, we counted them as dispersed. And we found that 19% of the juvenile mice that we put into the enclosures, which were down at equal frequencies of T and wild types, equal frequencies of males and females, that they actually crossed through this barrier. And T carriers crossed the barrier more often. They emigrated more. And more of them emigrated at high density, but when you take into account that there are more individuals in high density enclosures, so here the high density treatment had twice as many animals as the low density. We found that density did not have a significant effect on emigration, but genotype did. So now we have observational results from the barn, theoretical results, and now experimental results, all supporting a higher rate of dispersal of T heterozygous house mice. So to summarize, from 2002 to 2012, T frequency went from 1/3 to 0. We have very good evidence that sperm competition disadvantage reduced the fitness of T heterozygous males. The fitness of female carriers was not reduced. Higher population density increased female remating and therefore increased sperm competition. We found that T heterozygous mice are more likely to disperse, and this may explain why the T has not gone globally extinct. So we had local extinction in the barn and increased dispersal. So dispersal may have also explained some of the decline in frequency of the T. But these T carriers left the barn and presumably to search out populations of lower density where they would be likely to be more successful. And we've not found evidence of evolved resistance to drive. So we have no evidence of genetic suppressors of drive. So drive in the lab is still as strong as it was initially, and we've not found any behavioral types of resistance to drive. And currently, I'm working on two projects. One is to identify the proximate causes of increased dispersal of T heterozygous house mice. Why are they dispersing more? What's going on? Is it, are there some social factors that they're responding differently to compared to wild types? Does it have to do with physiological measures? Are they perhaps more anxious physiologically, and that causes them to respond differently? We're looking into what it might, trying to identify what it might be. And secondly, we're extending studies to another variant, the TW2 variant, which, when homozygous, causes male sterility. So females remain viable and fertile, but males are sterile. And this is an interesting variant to look at because it's been proposed as a means of driving SRY, the gene that determines maleness in mammals, with the idea of creating a highly male-biased sex ratio for population control of house mice on islands where they're causing conservation problems. And so it's a very interesting variant to study to better understand what, if it's might be a suitable system to work with. And so with that, I'd like to thank my funders, particularly the Swiss National Science Foundation, that has been very supportive of my work. And special thanks to Barbara Koenig, who started the study and has been a wonderful mentor throughout, and Ph.D. students who contributed particularly to this work, André Manzer, Andreas Sutter, and Jan-Niklas Runne, and as well technicians in the lab that have had strong, very helpful effects, including Jari Garbeli, Bruce Boatman, Gabi Stichel, and Sally Steinert. And I thank all of you for your attention as well. So thank you very much.
Thank you, Anna, for a wonderful talk, and really, really great and super clear and super interesting. Let me encourage those in the audience to feel free to ask questions. There are two ways that you can ask questions. You can either chat them in using the Zoom chat feature, or you can raise your hand and we'll recognize you and unmute your microphone. So go ahead and do that. I have a question. So I guess the explanation then, if I have interpreted what you've said, is that the extinction that you see in the barn was, well, at least it correlated with a rather large increase in population size within the barn. So do you think that that was the proximal cause of the extinction of T in the barn?
I do think it played a role, particularly through increasing multiple paternity. I think that's one of the key features, that the more mice there are, the more males a female encounters and the more often she mates.
Yeah. What's happening to the mouse population outside the barn? So presumably there's T out there now in the local population, but they're not coming back into the barn.
They're not coming back into the barn. So we have done a little bit of sampling, not very extensive. And we have not identified the T in the closest population, but we should really put more effort into discovering that. The nearest population that we know of is about 0.5 kilometer away. So it's probably not so easy for house mice to get there.
Okay. Got some questions in the chat here. Pratima Trinuri asked a question. First of all, hi, Pratima. Thank you. And she says, thank you for a very interesting talk. A crazy thought. Do carriers of t-haplotype have hormonal imbalances that make them more adventurous, reckless, where they are happy to cross the water barrier?
Well, we're hoping to find that out. So that's one of the things we'll be looking at, is also hormone levels. And hopefully that we'll be able to answer that in a couple of years.
Yeah. So not so crazy thought after all. So looking at hormone levels is actually on the agenda.
Yes, it is. Yeah.
Yeah. Super. Zhenjiang Chen asks a question. He says, great talk. Whether T-specific transcripts are synthetic to the genes on chromosome 17, and if not, what's the possible origin of T-specific transcripts? Okay.
So if I hope I understood the question correctly. So most of the transcripts were variants on wild type transcripts. So they were not completely novel, but there were several cases of transcripts that did not match the wild type transcripts on chromosome 17. But there still were a relatively small number of those. And we tested them, and there really were only these two that we could really confirm that had very closely related genes on other chromosomes and did have T-specific duplications. So we'll have to keep looking for those. An interesting thing about this study was that there's a good chance that there was more than one T variant included in the study. And if we looked, which will make it a bit less sensitive to finding these. And if we looked more carefully, I think within one T variant comparing to the wild type it's evolved with, I think we might find more. Does that answer the question?
Well, it sort of did for me. Hopefully, if Zhenjiang has a follow-up question, please follow up and with additional question if there is one. Hector Kamada asked to if you could elaborate a little bit more on that variant that you mentioned at the end, the T variant. Could you just describe that a bit more?
So there have been a fairly large number of different T variants have been described, and there are a small number that cause male sterility. And the TW2 was first found in New York back in the 40s and was kept in labs in the US for a number of years, and was studied quite a lot in the 1960s. And then it was faded a bit, and this particular variant of the T haplotype I have obtained from John Godwin in North Carolina, who has been working with it with Megan Sir, who got it from David Threadgill. So this is a lab variant that we've now crossed with our wild mice. So we have the TW2 against our wild mouse background, and we're starting to look at its effects on even looking at sperm production, what it's doing, and its behavior, and trying to understand aspects of the biology of the organism carrying the TW2 that we don't yet know about.
What's the nature of the variation?
So the research isn't really there. There's no genomic comparison, but research in the 1970s suggested that the MHC variants were different in these different variants. So they had independent evolutionary histories. They do have, these different variants have different lethal alleles. That's how they're defined, as being different variants. They have different, if you cross them against another T variant, they don't cause the lethal effect. So I think it's still quite understudied.
Yeah, so I guess I'm a little surprised that the genomics across that region, or the comparative genomics across that region. Well, it sounds like from what you just said that there hasn't been a lot of comparative genomics across that region. Is that true? And why would that be if that's so?
Well, one of the problems is that these inversions make it very difficult to sequence.
Yeah.
There's a lot of heterochromatic sequence, so there's degeneration in some aspects, and it all is hard to sequence.
Yeah.
So I do have a sequencing project with Jan Runge, and it's just been very hard to assemble the t-haplotype.
Yeah, we've heard similar difficulties from David Presgrave last week in terms of some of his work in dealing with repetitive elements, and also Amanda Laracuenta described in an earlier webinar her interest in satellite DNA and its role in segregation disorder in Drosophila, and how some of now the long read sequencing technologies have really opened up some possibilities for getting at getting at those regions. So I assume that should be helpful, maybe going down the road for for the T. I've got a couple more questions in the chat here. So Jose Hernandez says, thank you for a great talk. Is there any evidence of strong population size fluctuations in natural populations such that it could help explain the sustained frequency of T haplotype?
Well, we've seen population explosions happening in places like Australia. Population size reductions that are not caused by humans, I guess I'm less aware of. But house mice are very good at colonizing. They have colonized all over the world, and they can reproduce very well in low densities. So they can very quickly build up large populations. I guess we know less. Mice at low densities are harder to study, and we have less information about them.
A developmental biology question: what's known about the gene responsible for the lethality and the initial? I guess the initial phenotype was tailless, right? Is that where T comes from, right?
That's right.
Yeah. So, how much is known about the genes involved in that particular phenotype?
So there has been one gene has been identified as being responsible for the embryonic lethality in one of the T variants, but as far as I know, that's the only one. And we know that different variants induce lethality at different stages in embryonic development. So we think it's probably different genes.
Multiple genes.
Yeah.
Takashi Akira asks a question. He says, a naive question. Based on your finding, is it less likely that we will find T mice in smaller islands like Madeira where they cannot emigrate?
Possibly yes. Possibly yes. Yeah. It would probably depend on a lot of local conditions. There is not not so much data available to actually try to test this idea.
Good, thanks. Eric Lai has a talk. He says, thank you for a great talk and in history. I think I missed something basic from the beginning. Since T is autosomal, how is this haplotype recognized in the first place? And do you speculate that there could be other autosomal driver alleles that are not easily recognized without, without molecular confirmation?
So for the first question, it was, I mean, now it's fairly easy to identify the T haplotype. We can do PCR and test for it. But earlier, what researchers did was they crossed mice to a line that was heterozygous for the brachyury allele. And the combination of brachyury and T caused a different phenotype, then inheriting a wild type allele and the T. And so they had to do crosses and count phenotypes of offspring to figure out what the T frequency was. And this was very tedious. So just by chance, there was a phenotypic marker they could use. For the second question, which now I've forgotten. Please remind me, David.
Yeah, sure. And do you speculate that there could be other autosomal driver alleles that are not easily recognized without some molecular basis?
Certainly. I certainly expect more. And there have been some found in the literature that people have identified, but there hasn't been much more work done with. I certainly expect there to be more found with increased resolution of ability to find them.
Yeah. So I'm just going to ask you for some of your comments or thoughts on the conservation efforts that people are considering the use of gene drive systems for rodent control on islands for the purposes of biodiversity conservation. Do you first of all, are you involved in any of those efforts? And the second question is, have you thought about those kind of things, and have you had any opinions or ideas or thoughts on it?
I'm not currently involved with any such efforts. I am interested in island conservation and particularly conserving endangered species that are threatened by invasive rodents, including house mice. And the most popular method at the moment of using poison seems to be quite harmful and doesn't always work. And I think that there are new tools needed. And whether these new efforts turn into a tool that is safe and effective, I think I don't know yet, and we'll have to see what the tests show as things develop. I think we'll have to just keep an eye out for what's happening in that field.
Yeah. Well, great. Let's see. Januri has another comment here. She says, from an evolutionary perspective, does evolution come up with these selfish duplications to facilitate emigration, to sort of conduct its own experiment in nature, kind of stir the pot just to see what gives?
It is a little bit like sex chromosome evolution, where there are genes that, for example, benefit females that end up on the X chromosome. And there are some studies that have shown that there are transposons associated with some genes that allows them to to integrate at different regions of the genome. So from that point of view, these transposons might be enhancing the potential for the evolution of these T-specific behaviors. And I think I have to give it some more thought, I think.
Yep. Yeah, it was a speculative, certainly question. It was a deep one. Well, I'm not seeing any more questions from our audience. You're certainly free to continue to ask them, but you'll have to do it quickly because we're going to wrap up if you don't. In the meantime, what I would like to say is that next week we will continue this webinar series with our final webinar. And this particular webinar series, we'll have an additional series coming up later in April. But this particular series on selfish elements drive in natural systems is going to end next week with a webinar from Sarah Zanders, who will talk about some really interesting spore killers that she's been studying. So, Anna, I don't see any more questions. I think we've exhausted the questions from the audience. At this point, I think I'd like to say, thanks a lot for Super Talk, and thanks for taking the time late in your day to join us and give this webinar. We really, really appreciate it. And thanks again.
Thanks so much for the opportunity.
Yeah, great. With that, I'm going to close today's session or webinar. Again, thank you to the audience for attending. Again, thanks to Anna. Hope to see you next week for the final webinar in this particular series by Sarah Zanders and on spore killers. So have a nice day, everybody.